Nucleome Therapeutics is a biotech company that was spun out from Oxford University in 2019 with the goal of using human genetics to understand human disease. So we are focusing our work on inflammatory diseases. These are diseases where the immune system goes wrong and you get constant chronic inflammation. So think of inflammatory bowel disease, rheumatoid arthritis, lupus, the list goes on. And actually inflammation probably has some impact on almost every disease that we know. So this is an area where there's been a lot of progress actually in new medicines, including things that I've worked on over many years, but there's a huge amount still to do.
The problem in drug discovery is what we call translation. So it is understanding that the science you're doing in the lab is actually going to result in a drug that helps patients. Historically, people haven't looked in humans as a first step.
Key things about what we're doing is that everything we do is in humans. Mice and humans look like they're made out of the same thing. We're made of building blocks, proteins, that drive the way our bodies work. So if you think of the constituents as a pile of bricks, the pile of bricks looks essentially the same, but the mouse doesn't look anything like a human. The human genome is a sequence of about 3 billion so-called bases. These are the individual letters. At any point in those 3 billion bases, there may be alterations in the code that takes place. And some of those alterations are associated with the disease. We think that we can understand human disease looking at the mice because of the similarity of the bricks. And the difference actually is in the instructions for how the bricks are used.
Most of those changes in the DNA, they don't actually occur in genes. In fact, 90% or more of those mutations occur in what's called non-coding genetics.
The vast parts of the genome, 98% of the genome, does not actually encode directly the proteins. Encodes the instructions for how they're put together to create a mouse or a human, or a fly or a worm. All the variants that we look at are in these non-coding sequences.
They change the way that the proteins are made, the amount that are made. They may increase, they may decrease, and that's what's going to result in the disease.
Inflammatory reactions have a very predictable course. Something stimulates them and you get inflammation going up. But every inflammatory reaction contains the seeds of its own resolution. So turning off is as important as turning on. All of the drugs that we currently have actually block what are called the pro-inflammatory mediators on the up phase. They're not actually switching the thing off. They're just sort of catching the water as it comes out of the tap rather than turning the tap off. With the genetic approach, we can actually say, well, if we take a disease like rheumatoid arthritis, what genetic variant is associated with a higher risk of getting this disease? We find the variant, we find what gene is associated, and whether the gene is turned up or down. In a case where a protein is turned down, where you've got persistent inflammation, what that tells you is that that protein is actually involved in turning the inflammation off. It's a sort of double negative. So this looks like what we might call an inflammation checkpoint, something which naturally turns the inflammation off. So our lead program is one of these inflammation checkpoints. The protein we found was missing in patients who have a susceptibility to not only rheumatoid arthritis, but also systemic lupus erythematosus, SLE, probably inflammatory bowel disease as well. As I said before, all of the current anti-inflammatory drugs treat the up phase, not the down phase. There's a half the immune system, a whole half of the molecular biology of this system, which is untapped from a drug point of view, which we can see by asking the right questions with the genetics. We're in the process of making antibody-based drugs to target it, not by blocking things that are going on, but by stimulating the natural processes of resolution of inflammation.
Machine learning has effectively been critical to doing what we've been able to do. The core technology underlying our platform is a combination of two pieces that work in synergy together. So there's a machine learning technology and also a proprietary next generation sequencing laboratory technology. We focus on the machine learning first. What we do there is ask a very specific question, which is, of all of the human genetic variation that we can measure and associate with disease, which ones are the ones that are actually doing something functionally to a gene in a disease context.
A disease variant, a change in the DNA that is associated with the disease. And you map that to the gene of interest using our key technology, which is called MCC.
What that allows us to do effectively is focus our lab work on the things that are most likely to be effective. The second core technology is specifically asking the question of how do regions that are distal from each other on the genome in a linear sense interact when the genome folds in 3 dimensions.
So the essence of this ability to understand how all this genetic variation affects disease doesn't come through knowing the linear sequence of the genome, which is where most of the genomic data comes. It comes through understanding the 3-dimensional structure of the genome as it folds up in the cell. And it's actually the 3-dimensional structure which determines which genes are turned on and off, because the control switches, if you like, turn on and off the genes, and they assemble in space, but they may be very far apart on the linear sequence of the genome. This is pretty tricky because you've got the DNA in the nucleus of the cell. You have to kind of freeze the structure and then pull it apart in such a way that you can read which bits of the genome at this atomic scale are interfering with which, and whether they're on switches or off switches for the genes. And there have been technologies around for doing this for some time, but none of them have actually had the resolution you need to do the kinds of things that we want to do.
Our technology can do that precisely. We have effectively the most precise technology available at the moment. And critically, it can do it at scale.
We can actually see these control interactions which determine whether genes are going up and down. Then we look for control switches that have variations in them that are associated with the disease. So now this unlocks the whole thing.
So that allows us to link disease to variants to genes in specific cell types. And when we can do that genome-wide,
we now have information about multiple proteins that are involved. So what we can do is, instead of selecting a particular protein or a particular target, now we select a mechanism that's interesting. So this is fundamentally different from the way that most people do this. And if you do that, I can look at that mechanism, which might involve 20, 30 proteins, and I can say, this is the one that I want to go after, and it's involved in a really important mechanism.
We can start to ask questions effectively. What is the underlying mechanistic dysregulations? What is going wrong in autoimmune disease? How can we use this data is to move beyond starting to think about disease in terms of its mechanism rather than its symptoms. Take something like rheumatoid arthritis, and it isn't actually one disease when you think of it from a mechanistic point of view. It could be multiple mechanisms that drive the disease.
We start with human genetic variation. We find out what the molecular basis of the disease is. That allows us to pick the targets for drugs. And then we're actually using human cells and tissues to work out what the mechanism of action of those drugs is, which actually allows us to do things that haven't been possible before.
So this is really exciting. What we have been able to do is to take that technology and identify molecular targets that have never been identified before. We've done that with a target that we're calling NTP464, a target that will be potentially very useful in rheumatoid arthritis, but also in other autoimmune diseases, lupus and maybe inflammatory bowel disease. What we have found, by using our technology, you can affect a cellular and molecular mechanism that hasn't been previously addressed. This was a gene target that would be important in 3 different immune cells that are very important. They're called the B cells, which are important in generating antibodies. They're called the T cells, which are important in both suppressing autoimmune disease and fighting infection. And then dendritic cells, which are important for T cell function. Because we're affecting a very key mechanism, if you change those T regulatory cells, you normalize the immune system. I can restore normal immune function. The effect of the drugs that go after that target are going to be quite different from the effects of standard of care.
Moving the consideration of effectively heterogeneity in the patient population in a mechanistic sense to early in the drug discovery process, we believe is going to be massively beneficial in terms of discovery of novel therapeutics.
There is significant need for therapies in that area, and particularly a need for novel therapies. This platform has been very, very productive. The technology we have is broadly applicable. Our approach has identified 2 molecular targets that could be really important in multiple sclerosis, affecting different cell types there that we think could really have an impact on that disease.
We had a fantastic comment. You've got exciting data in a busy field. This is a solution to a problem that I've been worrying about for a very long time. I've been involved in half a dozen different programs over the years, and I was aware of what the uncertainties and unknowns were in biology of these diseases is a real challenge. To get at that complexity and resolve it into something that may be useful for creating medicines. It's actually why I love this business, because it's unsolved problems that we get to go into.